Chapter 14: Viruses, Cancer, and Immunology. Chapter outline: 14 to 1 viruses, 14 to 2 retroviruses, 14 to 3 the immune system, 14 to 4 cancer, 14 to 5 AIDS. Viruses are pathogens of bacteria, plants, and animals. Can be deadly, like Ebola and HIV. Can be merely annoying, like rhinovirus. Viruses are small particles composed of nucleic acid and protein. Entire particles known as virion. Capsid surround the center of virion. Nucleocapsid, combination of the nucleic acid and the capsid. Membrane envelope surrounds the nucleocapsid. Protein spikes help viruses attach themselves to the host cell. This is the architecture of a virus. Spikes, envelope, nucleic acid, capsomeres, capsid. This is the virus life cycle. Genetic material, DNA or RNA, entry of virus into cell, then transcription and replication, then translation, then count proteins, then cell replication, then assembly, and release from cell. Outcome of HD40 infection. In rodent cells, there's transformation, while the primate cells, there is virus production.
How does a virus infect a cell? A virus must attach to the host cell before it can penetrate. A common method of attachment involves the binding of one of the spike proteins on envelope of the virus to a specific receptor on the host cell. An example is HIV attachment. Retroviruses. A retrovirus implies that replication is backward compared to the central dogma of molecular biology. The genome of a retrovirus is single-stranded RNA. Once it infects the cell, the RNA strand is used as template to make double-stranded DNA. Retroviruses have been linked to cancer and AIDS. This is the life cycle of a retrovirus. Retroviruses have certain genes in common. Coat proteins, genes for proteins of the nucleoplasmid. All retroviruses have genes for reverse transcriptase or RT and for envelope proteins EP. This is a typical retrovirus genes. Summary of retroviruses. Retroviruses have a genome based on DNA. When they infect cells, their RNA is turned into DNA by RT. The DNA is then incorporated into the host DNA genome as a part of the replication cycle for the virus. Retroviruses all have certain genes in common. Some retroviruses also have identifiably unique genes. Example, the sarcoma oncogene and the rho sarcoma virus. Viruses are also used in gene therapy.
The immune system. The immune system allows for the distinction between self from non-self. This allows cells and molecules responsible for immunity to recognize and destroy pathogens. The immune system can also go in distinguishing self from non-self. This result is an autoimmune disease in which the immune system attacks the body's own tissues. Allergies are also another type of improper functioning of the system. Innate system. There are several parts to innate immunity: physical virus cells of immune system, dendritic cells, macrophage, and natural killer NK cells. Dendritic cells are members of a class of cells called antigen representing cells, APC. T cells release chemical called cytokines that stimulate other members of the immune system, e.g. killer T cells and B cells. Another important cell type in the innate immunity system in the natural killer NK cells, which is a type of leukocyte. Innate immunity. These cells differentiate and become specialized for one of several possible functions. Killer T cells involve T cell receptors, TCR, on their surface that recognize and bind to antigens. Proliferation of killer T cells is triggered when macrophage bound to cell produce small proteins called interleukins.
Clonal selection, the process by which only the cells that respond to a given antigen grow in preference to other T cells is called clonal selection. Macrophage T cell antigen, T cell growth, T cell. T cells are not bound to antigen, do not proliferate. How to helper T cells aid in the development of B cells? Response antigen to immature B cell to MHC2 to helper T cell. Process antigen binds to helper the T cells release into interleukins triggers proliferations of B cells. Next is to B cell. Maturating B cells can make the antibodies that bind to the antigens present in the bloodstream. B cells proliferate. Plasma cells release the antibodies into bloodstream. Antibodies. Antibodies are the Y-shaped molecules consisting of two identical heavy chains, the two identical light chains held together by disqualified bonds. Antibodies are glycoproteins. Heavy chains, light chains, disqualified bond, V domain binds to the antigen, to C domain activates complement and phagocytes and antibodies that are composed of antibodies. Antibodies contain, or the vertebral region is found at the prongs of the Y, and it's the part of the antibody that binds to the antigen. The bind sites for the antibody on the antigen are called epitopers. The pairs are composed of antibody and origin. Antibody heavy and light chains. This is the structure of antibody heavy and light chains. Heavy chains variable V domain, C domain to constant or DNA. To DNA to RNA. To heavy chain, light chains is covalent bond or antibody. C domain activates component and phagocytes. V domain binds to antigen to RNA. DNA to regrament, DNA regrament to constant DNA variable light chain. Antibodies contain it the cell's receptors encounter the self-antigens that are recognized with a highly affinity. It undergoes a process called negative selection and is the program for apoptosis or cell death. There are several safeguards that are leads to the delicate balance that must be maintained by the immune system. Different types of cell. This is the different types of cell. Thymus and periphery. Thymus is the positive selection slow affinity ligands. Cytosis T cell, helper T cell, self-C1 MHC self-peptide, self-C2 MHC self-peptide. The negative selection high affinity ligand is self-CT1 MHC self-peptide. So, self-C2 MCH self-peptide to apoptic death. Next is the periphery, survival low affinity, and activation high ligand. Survival low affinity is CDS to self-HC and self-peptide T cell CD4. The activation's high affinity ligand is CD8 to CD8, self-HC to foreign peptide to CD4 to CD4.
In summary, vertebrates have an immune system. Innate immunity consists of physical barriers and cellular warriors. Acquired immunity is based on two types of T cells and on B cells. These cells are generated randomly with receptors that can be specific for an unimaginable number of antigens. When cells encounter their specific antigens, they are then stimulated to multiply. Acquired immune cells have also leave behind memory cells so that if the same pathogen is seen again, the body is faster to eliminate it. Immune cells must be able to recognize self from non-self. T cells and B cells are conditioned not to recognize proteins from that individual. In some cases, the immune system breaks down and a person may be attacked by his or own immune system, leading to an autoimmune disease. And next, cancer. Cancer is the leading cause of death in human beings. It is characterized by cells that grow and divide out of control, often spreading to other tissues, causing them to become cancerous. All life-threatening cancers have at least 6 characteristics in common. 1. Cancer cells continue to grow and divide in situations in which normal cells do not. Second, cancer cells continue to grow even when the neighboring cells send out stop growth signals. Number 3. Cancer cells manage to keep going and avoid a self-destruct signal that usually occurs when DNA damage has occurred. And number 4, they can co-opt the body's vascular system, causing the growth of new blood vessels to supply the cancer cells with nutrients. Number 5, they are essentially immortal. And number 6, Cancer cells have the ability to break loose, travel to other parts of the body, and create new tumors, which make them lethal. This is called metastasis. Now, what causes cancer? Changes in DNA causes changes to specific proteins that are responsible for controlling the cell cycle. Most mutations of DNA affect two types of genes. Tumor suppressor, a gene that makes a protein that restricts the cell's ability to divide, and an oncogene is the one whose protein product stimulates growth and cell division. Mutations of an oncogene cause it to be permanently active. Now, for this slide, this is a table that represents a representative list of proto-oncogenes implicated in human tumors. So as for this page, the MAP kinase signal transduction. Signal transduction starts when a growth factor, the blue one, binds to a receptor monomer, the red one, on the cell membrane. The receptor is a tyrosine kinase, which then phosphorylates its partner receptor. The phosphorylated receptor is then recognized by GRB2, which binds to the RAS exchanger SAS. SAS is activated to exchange GDP for GTP on RAS. Activating it, RAS moves RAF to the cell membrane, where it becomes active. RAF phosphorylates MAP kinase, which then phosphorylates MAP kinase, the yellow one. MAP kinase, or NAPK, enters the nucleus and phosphorylates Jun, the light green one. The phosphorylated Jun then binds to Phos and CBP, and transcription is then activated.
But what is tumor suppression? Tumor suppressors inhibit transcription of genes that will cause increased replication. But what is tumor suppression? Tumor suppression is the DNA repair or the cell cycle repressors. It stops or regulates the overgrowth of cells that might lead to cancer. When a mutation occurs in any suppressor, replication and division become uncontrolled. A table is shown down below for the tumor suppressor gene and neoplasm connection. The action of P53. P53 is a tumor protein that regulates the cell cycle as a tumor suppressor. The mutations in the P53 gene are found in more than 50% of all human cancers. How do we fight cancer? It's been a decade and various research development has proved some various ways that cancer can be treated in a variety of ways, such as surgeries to remove tumors, radiation and chemotherapy, treatment with monoclonal antibodies to target specific tumors. More current foci include attempts to reactivate P53 in cancerous tissues when they have lost their function. This slide shows the drug targets in the P53 pathway. Transductional targeting in viral therapy is another way of treating or destroying cancer cells. In this way, a virus is injected to the body that can affect and kill malignant cells without harming normal cells while stimulating immune system and creating anti-tumor immunity. To summarize everything, all potentially fatal cancers have several things in common, such as having cells that are immortal that divide despite stopped growth, signals from the nervous cells that stimulate blood vessels formation near to themselves and that spread to other parts of the body. The development of cancer requires multiple breakdowns in normal metabolism. Such, most cancers have been linked to specific genes called oncogenes or tumor suppressor genes. When these genes mutated, the cell loses the ability to control its replication. There are many classical ways to fight cancer, such as radiation therapy and chemotherapy. Novel techniques using viruses are now being tried to target cancer cells more directly, and some of these are showing tremendous promise. That will be all the chapter 13 topic. Thank you.